Welcome back to Lab at Home with the Museum of Life and Science. I'm happy as always to be here with you along with Catalyst volunteer Caroline, who will be helping us with today's experiment. I hope that as always you will help lead our program with your thoughts and questions and ideas in the chat box. So this week we are continuing our Black Excellence in Science series to highlight the work and contributions of historical and current Black scientists, particularly Black scientists doing their amazing work right here in the Triangle. So today I'm really excited to share the work of molecular biologist, teacher, and science communicator, Dr. Ariana Eily. She wasn't able to attend this Lab at Home program, but she wanted to share more about her work and agreed to do a pre-recorded short question and answer session that we're gonna get to watch here in just a minute. It was really amazing to talk to her and hear about her work. Unfortunately, I needed to make a slightly shorter version of this interview for this program, but I will be posting the full interview on YouTube later in the week, and I encourage all of you to check that out so that you can hear more about her experience as a scientist, some really funny quirks of plant biology, and a very cool sounding science art exhibit that she was talking about seeing the world through the eyes of the sea anemone, like all kinds of cool stuff. I'm going to go ahead and show you just kind of the abbreviated version of this interview right now. So let's go and hear from Dr. Eily herself for this kind of quick Q&A before we do today's experiment. I'm going to go ahead and share my screen. Thank you so much for taking the time to talk with us today. I thought you might be able to introduce yourself better than I can. Could you tell us about some of the awesome things that you do?
Yeah, sure. I'm really happy to be here. So I am currently faculty at the North Carolina School of Science and Math. I teach in the biology department. And before that, I kind of got into science a little bit late in the game. As a kid, I grew up in a really rural area of Florida, so much so that they still herded cattle on horseback. And so my siblings and I played outside a ton. And so that kind of, exposure to the world around me is probably why I love science as much as I do. And, I won't give all of the details to connect the dots, but leaving high school, I decided to go to undergrad, and my advisor studied plants, and so he gave me some introduction into what research was like. So I researched carnivorous pitcher plants for a time, and then working with him is really what sparked me to make the choice to go to grad school. So during my time in grad school, I did my PhD through Duke's biology program and focused on plant bacterial symbioses. So how do plants and microorganisms live together, and in particular focused on these teeny tiny ferns and these cyanobacteria that they live in relationship with. But along the way, I also did things using improv to help scientists learn how to communicate and engage the public, which was really fun and continues to be really fun, as well as doing work in diversity and inclusion, all with kind of the goal of making science more accessible for everybody. So I think those things all kind of couple hand in hand really well. So I did my postdoc at Duke's Initiative for Science and Society, focused on that kind of aspect of science communication and science interdisciplinary work, all in ways to kind of deepen how science is connected to the world around us.
That's a lot of hats, as they say. That's a lot of things.
Luckily, I have a good head for hats.
You said that for your PhD, you studied some aspects of plant biology and cyanobacteria. And we're going to talk a little bit more in the program about what particularly cyanobacteria is. And we're going to be doing a project that's related to it. So I was wondering if you could tell us a little bit more about what you found in your research and kind of what you learned.
Yeah, absolutely. So my work was focused on these ferns named azolla, and a full-grown plant is small enough that it can fit on your thumbnail. That's as big as they're gonna get. Yeah, they're that bitty. And inside each of their leaves, they have this cavity that houses the cyanobacteria. And so cyanobacteria can do a whole host of different things in the environment. And the ones that are special in azolla actually do what's called nitrogen fixation. And so in the atmosphere, it's largely made up of nitrogen, and it's two nitrogen atoms triple bonded together, which is a super, super strong bond. And you and me can't break that bond. We don't have the molecular machinery to be able to do it. But these cyanobacteria do. And so they have this way of bringing in nitrogen from the atmosphere, busting apart that triple bond, and then turning that nitrogen into like things that you can eat and use and do other things that a cell might need to do with. And so in this symbiosis, they make nitrogen nutrients and they send it out to the plant. And in return, the plant photosynthesizes, makes sugars, and sends those sugars to the bacteria. So each of them feeds the other one. And so they've been living in this type of symbiosis for over 90 million years, which is like a crazy long amount of time. But we don't know how they communicate with one another. We were interested in figuring out, well, what things is the plant doing to support the life of the cyanobacteria and talk to it? And so I did experiments where we took the cyanobacteria away from the plant and compared that to plants that had their cyanobacteria to try and hunt down what genes are responsible in the plant for conducting this relationship with this bacteria. And then we also found different kind of chemical signaling mechanisms that plants produce a lot of for lots of different reasons that we think are involved in how they tell the cyanobacteria when it's time to make nitrogen, when it's not yet time to make nitrogen, etc. So it was really neat to kind of tap into something that is, you know, ancient and then trying to figure out kind of the secrets of what's going on there.
I love that you were kind of putting an ear to try and figure out, you know, these two organisms that live together. In fact, one of them lives inside of the other one. How do they talk to each other? And you also drew a connection between science and society and how communication happens between them. Can you tell us a little bit about that?
Yeah, absolutely. I think the two aspects of kind of how I think about science really merged together in the work that I did in thinking about communication between these two organisms and then thinking about communication between science and society. And in kind of thinking about that puzzle of how are science and society connected, the plant system that I studied also has all of these different like environmental impacts. So researchers are interested in it for creating biofuels so that we can replace gasoline, for combating climate change because it grows so quickly, it can pull CO2 out of the atmosphere really fast and has played a really critical role in the climate that we enjoy today, as well as being used in agricultural practices as a green fertilizer, so as a safe alternative to industrial fertilizers that get used instead. And so we have all these really neat, really cool environmental impacts, and the way that that is important is when you're able to share those findings about this plant with society so that it can go and be used for those purposes. And so in being a scientist, but then also in having all of these other interests in arts and humanities and how I used improv and did improv as a hobby and then used it for science communication, and then also being a minority in science myself, I just really got to thinking about access to science and who has access to science, both in terms of the exposure to it and the ability to use it in their daily lives and understand it, and who is doing science. And so I did a lot of work on kind of both fronts of helping prepare scientists to communicate and share their work and engage in outreach events like this one, where they get to share their work with people, but then also helping to create environments where minority students feel like they are supported and can excel in science, and kind of making space for folks who might want to ask different questions or have different talents and different skills that they can bring to science as a discipline. And in kind of thinking about how to create that space, and a thing that came out of it was the science and art exhibit that I started with some grad school friends. So we were focused on kind of science communication and science outreach, and we created this exhibit where we have artists and scientists working together to create pieces inspired by scientists' work, in large part because art connects to you in an emotional way. And so it gives you this different avenue to experience and start learning about science. And so it's been going on 4 years running now. It's called The Art of a Scientist, and it's just been a really awesome thing to create. When you'd mentioned before of like proud moments, it's something that I'm also really, really proud of just building this space where we have these two disciplines merged together and we get to learn from both of them is really, really special and a neat way to bring science and society closer together.
That is so cool. And I guess we might also say that both art and science kind of expand our perspectives. They show us things that we wouldn't be able to know or see otherwise.
Yes.
I love that. Dr. Eily, you have such a wonderful body of work and a wonderful way of looking at the world. And I want to thank you for sharing that with us today. And I guess I would ask if there's anything else you might want to share with our young scientist friends today.
Yeah, I would say first and foremost, I have gone on this journey and continued keeping the multidisciplinary things that I love a part of me, little bits of theater and art and science all woven together. So I'll never feel like you have to choose one if you love more than one. And we do have our next exhibit of The Art of a Scientist opening this spring. So it'll be a virtual experience. And so it'll open, we're being hosted by the Power Plant Gallery in Durham. So you'll be able to find it on their website come March. Yeah.
That's great. Thank you. I will make sure that I pass that on to them as well. And I'm really excited to see it. So I'll be on the lookout for it. Dr. Eily, thank you so much for talking with us today. And I really look forward to our own kind of deeper dive into some of these subjects. And thank you so much for showing us all that you have to offer. Thank you so much.
Thanks for having me, and enjoy making your cyanobacteria activities later.
Thank you so much. All right, I'll see you later. All right. Thanks so much for checking that out, friends, and thank you to Dr. Eily for doing that with us so that we got to hear from her. It's always coolest to hear about people's work from the people themselves. And she combines so many interesting things, and we talked a lot about a certain subject that we're going to go into a little bit more depth now so that we can do our experiment as well. When we were talking to Dr. Eily, she mentioned kind of the main subject of her study was something called cyanobacteria. And I was wondering, Caroline, if you could put that in the chat for us.
Yes.
Because we were talking about it, and I don't know if we kind of had a picture of it in our head, but cyanobacteria is a bacteria. It's a teeny tiny thing, kind of teeny tiny cells, and it lives in water, in basically all water. And where it gets its name from is its really cool ability to photosynthesize. And I was wondering if any of you have heard that term before. And if you have heard it before, photosynthesize, what does it mean? What does it mean to photosynthesize? You almost look like you're photosynthesizing right now, Caroline, with the beautiful sunlight.
I do. I'm sure that my potential cyanobacteria are very happy with this sun right here. Is this giving anyone ideas as to what photosynthesizing might be?
Oh, we have a friend who's learning about photosynthesis in science. Can you tell me some of what you remember? What do you think? Or maybe what does it have to do with? Oh, yeah. It means like turning air and sunlight into energy for a plant. Sun and water are needed. I love this. So we think about, and it makes glucose and oxygen. So it's taking some things and turning them into different things. And we usually think of photosynthesis being something that plants do. So plants like, sort of like the ones behind me, right? Have the ability to take the energy from the sun and take chemicals from the air and transform it into food for themselves. Exactly. These bacteria, instead of having to eat something else, like break down something else, they have the ability to eat sunlight and carbon dioxide in the same way. And just like our friend was saying, in exchange, they give things back. So they take those things and they make their food, and a byproduct of that is that they give off oxygen. And they also do stuff like helping to fix nitrogen, which we'll talk about in a second. But that oxygen part is really important. Cyanobacteria are, as far as we know, the oldest living thing on Earth. So they are some of the very, very oldest things. We're talking billions with a B years ago. They are definitely the oldest things that are fossilized, which is kind of the only way we have to be able to see what the world looked like back then. And those cyanobacteria and their process of photosynthesizing is what made our oxygen-rich atmosphere what it is today. So we wouldn't have all of the oxygen that we need to breathe if it weren't for cyanobacteria billions of years ago. So I think that's really amazing. They're kind of, in that way, maybe unsung heroes, but they do a lot of other things too. Some of those things we might consider being good, like they can be really beneficial to plants. Dr. Eily was saying that they can really help plants get what they need from the soil, like nitrogen. So sometimes plants need both that air, and they need sun, and they need water, and they also need a few other nutrients in the soil. Cyanobacteria helps some plants do that. Cyanobacteria can do other things, like there are sometimes big blooms of cyanobacteria in bodies of water. And sometimes cyanobacteria can make animals, including humans, sick. So it's not, I guess we couldn't say it's all beneficial to us, but it's part of our environment. And it's not one that we normally think of because it's so small. But since it's everywhere, I thought it would be cool for us to kind of do our own science/art, because as we've kind of talked about, they're really similar. We're going to set up an experiment to do with cyanobacteria. So we had you guys all gather some materials, and Caroline, I was wondering if you might share with us those materials, and if in the chat you're participating, I would love to hear if you are doing the experiment along with us. Go right ahead, Caroline.
Yes, definitely. We'd love to hear if you guys are doing it with us. Looking forward to finding out. But among the many things that we have today, we have water from some sort of source, be it a pond, a stream, any sort of kind of outdoor water that you know. Nice little jug of it like Peregrine has there. They're all ready to go. Also, you have some kind of detritus would be a term, which just is kind of dead stuff that has been logged in the water. I've got some sticks, and so does Peregrine. And then 2 containers with lids. My lids are over in the other room, but with lids ready to go. They don't have to be the same, but it just happens to be that both of us have the same size ones. Now, this other part would be a paintbrush and some glue. It's like school glue. If you don't have if you don't have school glue, like I don't, I actually have some tape that I'm going to be using. It's a good alternative. And then tin foil. So you can rip it, or you can cut it with scissors if your parents or guardians are around. But we don't need too fancy of designs. And then lastly, or at least I think it's the last thing, aside from scissors, would be a paper towel or regular towel just to keep things dry.
Definitely. Yes, exactly. Thank you, Caroline. And I just wanted to mention, somebody in the chat was saying that they're doing the experiment with us, and they even spent time playing in the creek today to collect all of the things that they needed. Creeks
That's
are
amazing.
Exactly. And creeks are a really great source of some of the stuff that we need. So in a moment, we're going to be putting our collected water, and it does need to be water from outside. It can't be water from the sink, because the water in the sink is treated so it's safe for us to drink. And the way that they treat it is to do things like kill bacteria. So if we want to encourage some bacteria to grow, which is kind of what our experiment is going to be, we're going to see if there are some things we can do, some factors we can add to our containers to encourage the growth of cyanobacteria. We can't use it from the sink because it has probably killed most of the cyanobacteria and other kinds of bacteria in there. So I have mine this is actually from the pond by the butterfly house. I made sure to get some of this here. And we were mentioning that we have some of the detritus, like you said, so some little pieces of sticks or leaves. These are going to be really important. But before we add those pieces, I want us to take a look at our containers, whatever kind of container you have. So I have these little guys, and instead of them having lids, I was just going to use some plastic wrap and rubber bands. All that needs to happen is to make sure that it's covered. But before we put anything in them, we want to do something to them first. And I'm going to go ahead and switch to my experiment cam so that we can take a look at what that's going to be. So I have my containers here, and I have my tinfoil. Tinfoil is one of those things that's a lab at home favorite. I just love to have tinfoil. It's really useful for a lot of things. One of the many things that it does is reflect light. And we know now that cyanobacteria needs light in order to do its thing, in order to make food, in order to make food and then make more of itself. So what this is going to do is it's going to help us take the light that will reach these containers and kind of bounce it back so it gets an extra boost of light. So what I'm going to do is I'm going to make sure that I can get the bottom of this covered in my tinfoil. Now that can look different for you. If you want, you can just kind of fold it over like that. What I am going to do is make a little bit of an imprint. So kind of made a little bit of a circle, and I'm going to cut that circle out. And this is so that whenever the sunlight comes into my bottle, it will hit this tinfoil that I'm going to affix to the bottom of my bottle. So I'm going to go ahead and I'm using glue. All we need to do for that is take some glue. I just have a little bit in here. And I'm going to paint the bottom of my container. Just like this. And now I'm going to attach this shiny side up. So the shiny side is pointing up, and all I'm doing is just putting it down on there. And I can use my hand to kind of smooth it on there. And now I have a shiny bottom, right? So my material is clear to let sunlight in, but now it has this tinfoil bottom that can bounce the light back. I'm going to do the same for this one. Something else with the tinfoil that I was thinking about when I was testing this idea is when I was putting it on the bottle, I just noticed that the way that it bounced light around looked really beautiful. And I was thinking about the connections between science and art, and thinking about how we could affect our experiment by making our experiment look more beautiful to us. So we can do what we need to do for our bottles, which is just to make the bottom of them shiny to increase the amount of sunlight for them. And we can do something else too. We can kind of take little pieces of tinfoil, and we can kind of create a wall that will help bounce light back into the container once more light has hit it. So I cut a few triangles that I could use. I also cut out this shape, which it kind of looks like a bunch of beads, and that's because I wanted it to look kind of like cyanobacteria, because when you look at cyanobacteria under a microscope, it looks just kind of like a bunch of circles. So I was thinking what I'm going to do is kind of attach that to my bottle. And whoops, the only thing I have to do that is paint a little bit of glue onto it. And you can see I'm not using a whole lot of glue because the tinfoil is really thin and it sticks really well to stuff. So I'm just kind of putting it on here. And then this would be my kind of my back. So when this needs sunlight, the sun will come in and bounce off of this shininess and then into my bottle, therefore giving more light to my hopefully future cyanobacteria in here. How are you doing on yours, Caroline?
I love it. Well, I am doing very well, doing similarly, putting a little piece of tape and ripping it in half, putting it along the sides to affix the tin foil to the bottom. Very easy to do again, don't need much tape. And now I have that on both. I also have a little artistic shape here, which is a little smiley face that I'm going to tape to the back.
That is so cute. That is so cute. So this doesn't have to look like anything in particular, or you can make it look like something. That's just kind of one of the many awesome things about an artistic kind of interpretation of our experiment here. This part is just meant to bounce some light around, and I'm sure that the shapes that we choose will affect the ultimate result, and we'll be able to see the evidence of that. So I've just kind of got some random triangles. This can look however you want. Something that I love about the connection between science and art, right, is that if you're an artist, you're also a scientist because you use the scientific method. You have to try things and experiment with them to see how they go. Sometimes things don't work the way you want them to, so you have to try something else. That's exactly what science is, collecting material, collecting information rather, in that way.
Definitely.
So I've got some of these here. And I've also got my cyanobacteria bottle this way. There we go. And you don't have to be finished putting the tinfoil on your bottle, but I'm going to show you just what we're going to do next. Once we do put all of our tinfoil on, the next step is just to get some of our liquid into our bottles. So what I'm going to do is I'm actually, since mine have kind of a thin, or rather a small opening, I was going to use a funnel to help me just because I have a really big container to pour from.
Opposite issue.
Right, yours is very streamlined, Caroline. So what we do now is I'm just going to pour in, I'm going to fill it up about 1/2 the way to 3/4 of the way full. And the reason why I'm doing that and not filling it up all the way is because I want there to be plenty of room for kind of the byproducts of photosynthesis. There's going to be gas exchange happening as the cyanobacteria eats. And since we're going to be sealing our bottles, we just want to make sure that we're giving a bit of room for the cyanobacteria and the gas exchange to kind of do their thing. So I think this is perfect for this bottle here. Now I'm going to fill up this bottle as well. Again, since this is just pouring, don't worry if you're not on this step. You can make your bottle look as beautiful as you would like it to.
Yes, take your time creating your art.
Definitely. All right. So I've got it about the same level here. Very nice. And one more thing before I'm going to seal the tops of these bottles. And that is, for one of them, I'm going to do something that I'm not doing in the other one. So one of them I'm going to leave without anything else in it. I'll just put the top of it onto it when I'm finished with it. And that's going to be, I'm going to choose this one for that. For this one, I'm going to do something different. I'm going to take my little sticks, and you can see mine's covered with a little bit of algae here, and I'm going to put it into my bottle. So if you have leaves, if you have sticks, if you have little bits of kind of soil, put them into one of your bottles, but not the other one. This is our variable. So we want to see, does it make a difference if I've got some dead sticks or leaves? Will we get more or less cyanobacteria or other effects? Or will it be the same as if we had nothing else in there? My hypothesis, I'm going to go ahead and switch to my face cam really quick here. My hypothesis is that that the one with some sticks in it, right, the one with some kind of dead material in it, is going to be helpful for cyanobacteria because, kind of like plants do, many plants do, they need other things than just water and air and sunlight. They need a little bit of mineral to kind of help them. And I know that as things like sticks and leaves start to break down and decay, they release phosphorus, they release nitrogen, and all of those things are things that cyanobacteria, that really helps it grow. It's something that it likes to eat. So I'll be interested to see the difference. All I need to do now is put these in front of a windowsill. And it always takes, like all good things, it does take some time. But after a few weeks, I'll show you this other one I started growing. It grew a whole lot of stuff. Now, I think that probably the majority of this is just some algae, and algae is a plant, even though confusingly cyanobacteria is sometimes called blue-green algae. Cyanobacteria is not a plant, but I think most of this is. But I did notice a few things kind of floating at the top, and sometimes little kind of colonies of cyanobacteria will float to the top. So regardless, it was cool to grow things, and I do think that my sticks in here did help help this algae proliferate to grow like it did. This only took a couple weeks because all of this stuff just likes still water and lots of sunlight. So I'm really interested to see how mine look. Caroline, yours look really beautiful as well. I like that you have that beautiful stick in there.
Yes, a nice little stick. And we've got our little backside triangles and smileys. I'm very interested to see how they will grow as well.
Yeah, I agree. I wanted to show you all one last thing, and that is we've been talking about things that are a lot of times until they've grown large enough, they're hard to see with just our eyes. So I have a little digital microscope that I wanted to use here just to help us take a closer look at some of the water, because I know that, I don't know about you, but when I'm looking at this water, it just looks kind of cloudy. It's hard for me to think about there potentially being life inside of here.
Definitely.
So what I'm gonna do is I am going to kind of look at some different little bits of I have a little bit of my water here and some of the sediment that was on the bottom of this jar. I also just wanted to take a closer look at some of that algae. So I pulled out a little bit of that algae. We'll take a closer look at that. And then I was just gonna take a ladle and take a look at some of the water that I collected in the butterfly house pond, just to see what that looked like under the microscope, to give us kind of a better look. And I don't know if you tuned in for previous labs at home, but we have done a little water drop microscope, so that might be able to kind of help show some of the things that we're going to see here in just a second. I'm going to switch to my microscope here. The first thing that I'm going to look at is that algae, because it just looks so neat. So here we are looking at some of the algae that was growing. Again, not cyanobacteria, but I mean, it's cool, it's green. A lot of times algae and cyanobacteria kind of grow together, or they compete for resources. So it was really cool to see that, and you can see there's lots of things inside of the algae as well, like little pieces that are trapped in there. I wanted to show, this is kind of, when we're talking about our sticks or our leaves breaking down, right? When they start to break down, a lot of times it looks kind of like this. So this is the kind of stuff we're looking at really closely to see if this affects how our bottles may change or grow, whether that encourages cyanobacteria or anything else to grow in it. So that looked really cool. And that came from having some decaying organic stuff inside of the jar. So the last step of this is just kind of covering the tops. And that's just to kind of keep the water from evaporating, so the same amount of water keeps in here, and to put it by a windowsill or by a window so that it gets plenty of sunlight. You can put them outside, but I bet they might like it even better inside of your house where it's a little bit warmer. So that's what I would recommend doing. We've kind of created our own little, hopefully future cyanobacteria gallery. Even if it doesn't end up growing cyanobacteria and it grows more stuff like algae, it'll still be cool to look at. And I like the idea of putting it by the window, because then maybe other people could look at it too. Sounds like some of us have made ours already. I can't wait to see how those look. Always feel free to share those pictures with us if you want to post them on social media and tag the museum. That's always something that we love to see. You can also send them to our email address that's kind of attached to this Zoom. And good luck growing. I know that I'm interested in seeing my growing and seeing your growing, Caroline. That is the time we have for today. Next time, we are going to continue / conclude our celebration of Black excellence in science with a really cool series of experiments and kind of like games to do with neuroscience, to do with the brain. This is going to be really, really fun, and I hope that I can see you all then. Thank you all so much for checking this out with us. I hope that you have a great rest of your day. Thanks so much, Caroline, for helping us with this experiment.
Of course, glad to be here. Thanks everyone for joining in.
For sure, and we'll see you guys next time.
Bye.